Muy buenas tardes a todos. Mi nombre es Camilo Ramírez. Yo soy profesor del Instituto de Biología en la Facultad de Ciencias Exactas y Naturales de nuestra Universidad de Antioquia. Yo coordino el grupo de investigación en Biotecnología, Microbiología Agroambiental, Bioma, que está adscrito a mi dependencia obviamente y también a la Escuela de Microbiología, que activamente nos ayudó pues a organizar y a difundir este evento. Vamos entonces a dar comienzo a la conferencia con el título pues que ven en pantalla, dedicada a la presentación de la técnica conocida como Droplet Digital PCR, que es una cuantificación. La utilizó nuestro conferencista principalmente para la evaluación, la cuantificación de colonización de microorganismos endófitos en una planta de importancia para la producción de bioenergía en los Estados Unidos. Bueno, el doctor Robert Tournay, que es nuestro conferencista invitado, es un asociado postdoctoral de la Universidad de Washington en Seattle, quien está de paso por Medellín y por nuestro grupo de investigación en algunos trabajos también de colaboración científica con nosotros. Es para mí pues un placer tener a Robert en la conexión. Y bueno, Robert, we can hear you.
Ok, gracias. Disculpe, pero voy a dar la charla en inglés, porque aunque yo sigo aprendiendo español, es un poco, no creo que es suficiente para algo así. Entonces voy a cambiar a inglés. Ok. So I'm going to talk a little bit about, as Camilo said, our research, specifically with ddPCR, so droplet digital PCR, and strain-specific primers for our work with endophytes in the bioenergy of Poplar. So I've broken this down into three sections today. The first section, I'm going to give a little background and kind of my advocacy for environmental microbiology. And then I will change into showing you a little bit of the prior work that we've done in our lab so you can get an idea of what we do. And then we'll finish off with our digital droplet PCR experiments. I know how it is. I'm not sure on the English level on the other side, but I know how it is to try and listen and learn in another language. So, and I also know that I talk pretty quickly, especially when I get going. So I'll try and keep it slow, but I would be completely comfortable if anybody just wanted to jump in and ask a question. I might ask Camilo to kind of keep an eye on the chat for me. Okay? If you pop a question in there, we'll get to it. So let me, first, I'm going to try and minimize any kind of jargon or technical language or explain it as I go. But one thing that I'm certain, or two things that I'm certainly going to do is I'm going to use the term strain, which is refers to members of the same species of bacteria, but that have some genetic differences. And so there are enough differences between the two strains that they are classified as different than each other. The second thing that I'm going to do, I don't remember what it is. I lost it. It'll come back to me. Anyways. Okay, so I think for a lot of people, at least the general public, when they hear about bacteria, the slide kind of demonstrates what comes to mind, right? So the myth is that all bacteria are bad, and the myth is that that sterile is better, right? And this comes out in our marketing and our advertisements. And these are just some examples I pulled off of. I tried to grab Colombian brands, just an idea of how pervasive kind of this message or this image is of bacteria. And so bacteria are very good at adapting to their environment and changing. They have very short generation times. A single colony can contain billions of cells, and so the rates of mutation or generation of new mutants is really high. And also they can acquire DNA from their environment, either through eDNA or through horizontal gene transfer, the exchange of genetic information between two cells. And so the combination of these two factors, our perception or our idea that bacteria are bad and that we need to sterilize everything, in combination with their adaptability and ability to evolve quickly, has created a lot of problems with antibiotic resistance. And I'm fairly certain that most people are generally aware of the issue here, and so I'm not going to go through each of these, but just to give you an idea of some of the things that are coming out in the media and out in the research regarding antibiotic resistance. So really the spectrum of host microbe interactions is that most microbes are neutral, and that a few are pathogenic and a few are beneficial. And I remembered the other thing that I was going to try and refer to. So we work mainly with bacteria. So I'm going to use the term bacteria, and I'm going to use the term strain a lot. But a lot of what I'm saying applies to other microorganisms within the microbiome, right? So fungi and other microorganisms. So bacteria are involved in every major biological process or biogeochemical process on Earth. They're really the drivers of life and the exchange of chemicals and elements through the ecosystem. They're really the interface between the biotic, the living portion of our biosphere, and the abiotic, the non-living portion. They, of course, generate a large amount of oxygen. They accelerate the release of minerals into the environment that then become bioavailable for other organisms. They're a large part of the productivity in the ocean. They are the master decomposers. They make nutrients available for other organisms, whether it's fixing nitrogen or breaking down organic matter or releasing inorganic elements from stone and rock. So this is from a paper on weans, and he was reviewing a paper from 2011 by Mora, where the context was how many eukaryotic species are there on Earth, right? And so you can see in the second column is as near as I could find the most current estimates of the number of described species we have by these 5 major categories. The second column, the estimated species richness, is what Mora presented in his paper at the time, of what he thought that there was for, or what they thought that there was for the number of species on Earth. And then the last column is some other estimates from other articles as far as how many species, how much diversity there is on Earth. So I've added in bacteria on the bottom, and the current number that I can find is that we've described 21,000 species, where Mora predicted at that time in 2011 that there was around an estimation of 9,700 species. There's been literature that tries to put the number between as low as 2.4 million, and there's actually a paper that has estimated 3.2 trillion species of bacteria. So this paper, I think, is a little more of a reasonable estimate, and it falls in around 1 billion and 6 billion, which is still a pretty hefty number. But one figure that I really liked from the paper was comparing the estimates of life over time. And the 1992 article is actually E.O. Wilson. And you can see I've highlighted in the box that the estimate at that time was 0.4% of species were bacteria. And then Mora actually was a little less, 0.1. This study is estimating bacteria, and you can see the shift in the relative number of species has dramatically gone toward bacteria. So even less on the genetic diversity in bacteria. These two articles here are a pair of related articles. The first one came out in 2021, that's the Blackwell. And what they were asking was how much do we know about bacterial genomics? And so they collected all of the assembled genomes from the databases like NCBI and EMBL, and then they assembled an additional 300,000 genomes from short read archives and other sources. So looking at those 661,000 genomes, only 2,300 species were represented by those 63,000. 90% of which were 20 different species, all of which, or nearly all of which, are clinical strains. So the update to that, and this is actually a database that gets updated periodically called All the Bacteria, has now taken the number of genomes up to 2.4 million, combined with the original 600,000 and an additional 1.7 million that they sequenced, which now represents a little under the 12,000 species. Still, those same 20 clinical strains comprise more than 90% of the total genomes. So everything that we know about bacterial genetics comes from very few genomes and majority of strains or bacteria that interact with humans, mainly as pathogens. So in terms of environmental microbiology, we know very little about what's out there. Excuse me one second, I'm gonna grab my water. So we see this in our lab. So since 2022, we've sequenced and classified 95 strains of bacteria. And one thing that I am interested in and watching and noted is that the proportion of hypothetical proteins is much higher in environmental strains than the clinical strains, the well-studied ones. So the top 5 are just 5 examples from our library. The second column, the coding DNA sequences, those are, when we annotate the genome, those are the regions of the genome that look like proteins. They have the pattern of a protein, they have start codon, stop codon. And from that, a subset is broken down into proteins with known functions. And then there's a third group, or a second group, excuse me, of the hypothetical proteins. So they look like proteins, they have the characteristics of proteins, but there's no known function. So in our environmental strains, the proportion of hypothetical proteins runs 20, 30, 40% of the genome. Also, and sorry, I forgot to mention this, most of the genomes that we sequence, the new strains, are novel or potentially novel species. So as much as 2/3. So not only are the species unknown, but the genes that are underlying their genomes are not well known either. And you can see I just pulled 3 common clinical strains, E. coli, Pseudomonas aeruginosa, and Klebsiella. And you can see that the number of hypothetical proteins is much lower. They're much more studied. Okay, so this is my shift. That was my background. So now I'm going to move on to some background on our lab and what we study to set up what I want to talk about in the final third. Is there any questions so far, comments? Okay, I'm gonna move on. Okay, so most people are aware of of the human microbiome and the role of, or at least aware of the human microbiome, and that we have bacteria, fungi, viruses, protists that we carry with us that colonize the internal and external spaces on our body. The diversity of that, or the quantity of that, estimates of how many bacterial cells or how many microorganism cells, microbial cells we carry, has ranged as high as 10 times. So for every human cell that you have, the current estimate is that you have one microbial cell. So we have about 30 trillion human cells. That means our microbiome is estimated to be 30 trillion microbial cells. That difference is much greater at the genetic level, and I see estimates is of 100 to 1 is pretty conservative. There are papers showing 400 to 1, 500 to 1, 800 to 1. So from a genetic diversity, we carry more genes from microbes than we carry from humans with us. I think I saw a chat there. Camilo, when you get a chance, can you check that for me? Just make sure.
I'm checking out.
Okay. Okay, good. Okay. So plants have an analogous system, right? So they have microorganisms that colonize their internal and external tissues. And I've sort of, with my handy little pen here, tried to identify kind of the general compartments for the plant microbiome. The phylosphere being above ground, exterior tissues, the endosphere being interior tissues, both above ground and below ground. Most people are familiar with the rhizosphere. That's the most well-studied component. And so that's the soil or the area immediately around the roots. And then like the anthrosphere and carposphere are the flowers and fruits. So our lab, we focus on the endophytes. So the microorganisms, primarily bacteria that colonize the internal tissues of plants. And then we focus on a subset of that, the beneficial ones, the ones that promote host tolerance to different abiotic and biotic stresses. And they can do, these endophytes can do a wide range of, a wide range of activities for their host. So they can help acquire nutrients such as nitrogen and phosphate and iron, nitrogen fixing being kind of the most well-known, right? They also synthesize phytohormones. So hormones that are used by the plants, they have no known biological or metabolical use for the bacteria themselves, but they produce these hormones that then benefit the host. So for example, if you have endophytes that are colonizing the root and they're producing auxins, which can enhance root growth, then the host benefits by having more access to nutrients in the soils, and the endophyte benefits by having a host that can take up more nutrients from the soils. They also synthesize antimicrobial compounds so they can act against pathogens, either through the antimicrobial compounds or just generally competition. And then they can reduce host plant stress responses. So for example, ACC deaminase cleaves a precursor to ethylene, which is a plant stress hormone. And the ethylene for plants is a little bit like Goldilocks, the story. Too much, or a little bit is good, too much is not good. And so by cleaving the precursor to ethylene, the endophytes can lower the stress response or the ultimate ethylene response in the host. So we are exploring applications for these beneficial endophytes for things such as poor nutrient soils, suppression of pathogens, increased drought or heat tolerance, and then resistance to toxins. So for example, my PhD was in phytoremediation, which is using plants to clean pollutants out of soils. And so I was investigating the role of endophytes in increasing host plant tolerance to arsenic. And then just generally increased growth and vigor. So the endophytes can interact directly with the stresses, such as in the case of pathogens or in the case of toxins, or they can just generally make the host more healthy and more resistant on their own. So these are some examples. I'm not going to spend a lot of time on them. I just want to give you an idea of our past research. So I started with a yeast for Camilo. So I think it's our only yeast that we actively work with. And you can see the difference. These are pepper plants that were inoculated with a single strain, so just the WP1. The difference in both biomass and yield is apparent looking at the photos. These were grown under low nitrogen conditions. So it also is a phenomenon that we observe in grasses and conifers. So you can see on the left, the grasses were inoculated with a Burkhold area. There was increased total nitrogen in the above ground tissues and increased biomass overall. And it's pretty evident there in the roots as well. And then on the right with the conifers, that's a big, well, our forests are conifers where I live, and then it's also a big commercial product. They harvest the timber or the trees for timber. These plants here were inoculated with a consortium of 8 strains. So WP1 is the yeast, and then the rest are bacteria. And you can see the difference in the size and biomass of these Douglas fir seedlings. Okay, so this is phytoremediation. This is a project that we worked on at the NASA Ames site, and it's a Superfund site. And in the US, our most polluted sites are classified as Superfund sites. That means that they have lots of often many different kinds of toxic chemicals, and at high levels, and in close proximity to people, so that there's a high risk of the people around these sites coming into contact with these chemicals. So these Superfund sites often qualify for federal funding, or at least they used to get cleaned up. Okay, so the primary contaminant on this site was trichloroethylene, which is an organic solvent that is used often as, I think, a degreaser, and it's one of the most prevalent organic pollutants in the US. The trees that we see are poplar, Populus trichocarpa, so black cottonwood is the local term for us in the Pacific Northwest. These trees are clones of each other. They're inoculated with a single strain, our PDN3, which is sort of our superstar phytoremediation strain. And you can see that you can see a dramatic difference between the controls, the uninoculated trees, and the inoculated trees. And then we saw substantial reductions in the TCE concentration between the TCE was moving through the ground in the groundwater, and so they were measuring it above the site and below the site. The nice thing about poplars, and one of the reasons that we favor them, is they are, besides the fact that they're a local tree, they have a very broad habitat range. They have deep, extensive root systems, so their roots will penetrate meters into the ground. They grow very quickly, meters a year. And you can coppice them, meaning you can cut them off at the base, and they'll regrow vegetatively the next year. And then we also have done some work against fungal pathogens. So these are some examples of some dual plate assays on some agriculturally important fungal pathogens. And you can see the zone of inhibition created by the bacterial colonies as the fungal pathogen grows out from a plug in the center. The bacteria are producing a compound which inhibits the growth of the fungal pathogens. These are from 2 papers, and they're all Burkhold area. Okay, so now I'm kind of shifting into the reason for our recent work with Droplet digital PCR and strain-specific primers. So we're all digital here, so I won't wait for responses, but the idea here is which of these plants were inoculated and uninoculated, and they're from the same experiment. The top group is exposed to water stress, and you can see that in these photos in all 4 plants. Bottom group was the control, no water stress. Oops, that did not work. That did not work. Oh, it's my PDF, that's why. So behind this was a box that was going to pop up that says none of these were inoculated. No, excuse me, all of these were inoculated. So these are all inoculated plants, all 4 plants in both groups. And so one of the challenges that we have these great looking results in the plants and crops that we work with, but you can't see the microbes, so the evidence becomes somewhat circumstantial. We can say when the endophytes are there, the plants do better for whatever experiment, whatever stress. When they're not there, they don't. But what we've arrived at is that we need a line of evidence that we can demonstrate that the endophytes have successfully colonized the plants used in the experiments. Our second problem, and again, I was going to ask a question, but the idea here is we see differences in the distribution of the data between the inoculated plants, which I've highlighted boxes in red. These are draft plots from a heat tolerance experiment that we're writing up now. So final mass growth and then chlorophyll metrics. And you can see that the distribution of the inoculated plants is generally much greater than the distribution of the uninoculated plants. And we see this phenomenon through different biometrics. So either physiological response, such as growth, biochemical response, chlorophyll production. We're seeing it now that we're doing more metabolite studies. We see this much greater variation in the data between the inoculated and the uninoculated. Camilo, you have any idea why? You have a guess? I'm gonna pick on Camilo because he's here.
Maybe because some of them are responding.
Yeah, so it could be a response to the plants, or the plants interacting with the endophytes. My hypothesis is that we're getting uneven colonization. So some plants are getting colonized really well, others not so much. Maybe some plants not getting colonized at all. So that's our two problems: is being able to demonstrate conclusively that the endophytes are in the plant, and that the benefits that we're seeing, the results that we're seeing, can be attributed or more closely correlated with the presence of the endophytes. And the second, trying to explain this observation that we're seeing consistently across our experiments in the much broader distribution of the inoculated plants. So to kind of give some context to that, this is how we prepare or how we inoculate our plants. It's super high tech. We grow the endophyte cultures, we suspend them in a media, and we soak the roots in the inoculum for 24 hours. The plants are, with the poplars, are genetic clones. And so they're all very similar genetically. And then from here, once we have colonized them for 24 hours, they are then used in the experiments. So what I'm looking at or became interested in, I'm calling the colonization dynamics. So what we want to know is, are they successfully colonizing the plant? If they are they localizing? Where are they localizing? Are they staying in the roots, or are they moving to the shoots? And then, is there abundance? Are they colonizing at a high enough abundance to be biologically relevant? Okay, so now I'm going to transition over to the ddPCR experiment. So this has been kind of a setup, talking about the importance of environmental microbiology, and then a background on our experiments and the results we're seeing and kind of what led us to this type of research. Okay, so bioenergy crops right now almost exclusively come from seed crops, corn, soybean, and oil palm, and only the oils that we can extract out of the seeds. Lots of energy in these triglycerides, lots of energy in these oils, but the amount of biomass that's grown relative to the energy that we're capturing out of them, a lot of that, the whole rest of the plant is wasted. And these crops compete for agricultural land, displacing corn for people, soybeans for people, oil palm for other products for food too as well. And so it creates this dynamic of a low return on the amount of biomass in conjunction with the competition for agricultural lands. So there are two groups, University of Washington and another one at the Washington State University, that are working on the technology to convert cellulosic materials and the entire biomass over to ethanol. So to break those bonds down between lignin and cellulose that are hard to break but still store energy, so that we can convert energy, or we can create energy or biofuels out of virtually any carbon source. Our group at the University of Washington is working with poplars. For a lot of the reasons I mentioned before. They grow quickly, they have extensive root systems, you can coppice them and reharvest them again the next year. It has other benefits, ecosystem services like carbon capture and habitat, features like that. Additionally, they don't need to be grown on agricultural lands. They can be grown on any soils, including ones that are mildly contaminated. One of our research projects, or as I mentioned before, is looking at the role of poplars and the ability to clean pollutants out of soils. And so you can kind of close that circle of having a product that can be taken to market in the biofuels, but then also potentially could be remediating or cleaning up contaminated soils. Now, our role in that is to use our endophytes to make the plants more healthy and resistant to these environmental stresses. Primarily in this research, nitrogen and then heat or drought tolerance. So another challenge that we have is that we don't know exactly where our endophytes, or if they're in the plant or where they have colonized the plant. So on the left here is an example of one of our field poplar trees that we're collecting samples from, and you can see that it's 4 or 5 meters tall, and that happens every summer because we coppice them in the winter and they grow back over the growing season in the summer. So we can't see the microbes. We can't test the whole plant. Additionally, there is a host microbiome that contains dozens or hundreds of bacterial strains, some of which are the same species or very closely related to our own strains. And so it becomes sort of I don't know if this saying translates down here, but we would say a needle in a haystack. You're trying to find a very small or rare target with a lot of noise, a lot of noise in the background. And so we know that they're not evenly distributed. Life in general tends to cluster around resource-dense areas, right? So my example being the Nile River, the Great Barrier Reef, and then of course humans in Europe, we tend to cluster around, living things tend to cluster around resources. So it's not like I can just pick one leaf, sample that, and determine whether there's endophytes in the plant or not. Additionally, when we extract the DNA, we only load the tubes with about 50 milligrams of tissue. So there's a 4 meter tall tree that I can sample 50 milligrams of tissue out of. So the first part of this solution is droplet digital PCR, ddPCR. So on the left is an example that I'm sure most of you have probably seen of standard traditional PCR, right? Which uses a combination of chemicals and a heat-tolerant polymerase to copy target and make many copies of a specific DNA sequence. Okay. However, there are limitations in standard PCR that don't work well with our project. First of all, you need high concentrations of the target. Standard PCR is very sensitive to contaminants that inhibit the PCR reaction. And it's more qualitative than quantitative. There are ways to quantify PCR, but we can't get down to the resolution that we can with droplet digital PCR. Okay. And then extracting DNA from plants, I'll say humbly, is probably the most difficult substrate to extract DNA from. Maybe ancient prehistoric DNA from fossils, maybe. But plants are just challenging. Every plant is different. Every tissue is different. The age of the leaf, the root compounds, fine roots, heavy roots, they're all different, and they're very challenging, and they're full of inhibitors, polysaccharides, phenols, carbohydrates that inhibit the PCR process. It's hard to get a high concentration, pure sample of DNA from plants consistently. So secondarily, our target is the definition of rare, right? So 99% of the DNA that we extract is host DNA. The other 1%, the estimate is 1% is the microbiome DNA, of which our endophyte strains are a fraction of that. So we have a very noisy system and a very rare target. And again, as I mentioned before, and we can only process very small pieces of the plant per extraction. So Droplet digital, or ddPCR, the next is the second generation PCR. It takes the PCR reaction and samples or subsamples it into 20,000 droplets that are immersed in an oil. And then it performs 20,000 simultaneous PCR reactions on that one sample. In the super mix is a dye that bonds to DNA and that fluoresces. And so after the PCR reaction, we put it in a third machine. So there's a machine that creates the droplets, then there's your PCR machine, and then there's a machine that reads the fluorescent of the droplets. And so it feeds the droplets through one at a time and measures the fluorescence. And because it partitions the sample into such small aliquots or such small samples, it's much more able to pick up rare targets. So the resolution is really good. And it's a lot less sensitive to inhibitors because the inhibitors get diluted between the droplets. So down on the left, you can see three examples. The first one being no positive droplets, then low, medium, and high concentrations of positive droplets. And so based on the number of positive droplets, and because each droplet is a known volume, they can use Poisson statistics to estimate the number of copies of the target down to a very low level. So the dynamic range of ddPCR is 1 to 120,000 targets per reaction. So 22 microliter reaction, they can detect down to a quarter of a target per microliter. So very low numbers. As I said, it's resistant to inhibitors and non-target DNA. We have lots of noise in our samples. And we can get good estimates of the absolute quantification of the number of targets. So this is the output of, this is actually an optimization experiment. And you can see the positive droplets are blue. The negative droplets on the bottom are gray. I've got a control in the left column, and then I have a high concentration, low concentration, high concentration, low concentration of two of our strains. And so you can visually see on the screen the difference between the positive droplets and the negative droplets, and then visually see differences in the number of copies. And then it produces a spreadsheet that has this information for us. So this is really just for show. And I do have to say that they don't always look this clean. These droplets in between the two are what's called rain, and those are partially amplified. So they're just basically noise in the system. And so we have to figure out kind of where to place that threshold line in order to estimate the concentration. Okay, so getting back to our highly sensitive targets. So we've got a machine that can read them and at very low concentrations. Our second challenge is that a lot of the endophytes that we work with are very common plant-associated endophytes. And so there will be lots of strains in there that are very genetically similar to our strain. So it becomes challenging to design primers that will target our strain, but not target the closely related strains. So I don't know how, do you know what Waldo is? Where's Waldo? Ah, okay. A little cultural gap here. I'll just explain it really quick. It's a kid's book. It's super famous in the US. Normally the pictures they'll be just kind of a background scene, at the beach or at the mall, and there'll be one Waldo kind of hidden in there. And so the parents will sit down with their kids and they'll look for the Waldo. And so that series of books is called Where's Waldo? Right? So I've used ChatGPT here to make images of a microbiome with lots of Waldos in it. So the one on the left, they're all the same Pseudomonas species. What we're looking at is the image on the right, where there's a lot of Waldos that look like our strain, but are slightly different, the blue cap. Okay. I thought for sure that it would translate. All right. Okay, so strain-specific primers. So even though strain A and strain B, the same species, are very genetically similar, there are differences. We just have to find them. If we have a unique sequence of DNA from our strains, then we can then design the primers that target that sequence that we can then amplify with PCR and read in the Droplet reader. So the method that we do for this is we have the genome sequenced. This is all done in computer, in silico. And my two examples here are two pseudomoni. Step one, we have the genome. Step two, we use a small script by one of the researchers at JGI, which is one of our national labs, that segments the genome into 300 base pair segments in silico. So the 6 million base pair genome, I ended up with 20,148 segments, 300 base pairs long. And with the other one, about the same. Then we blast that. We run it against an algorithm that actually was designed to look for DNA sequences that have homology, that are similar in sequence. But what we can do is we can take the rejects. So it will go through 21,607 segments, and it will compare it to a database of 2,000 Pseudomonas genomes that we downloaded from the NCBI, and then categorize them as matches or no matches, and we keep the ones that are no matches. It doesn't always get down this far this fast, and so often what we'll do is just run it against other databases of closely related genera of closely related genera until we get a small enough number that becomes manageable. Right? And it doesn't matter, I could run it against any genomic sequence. All we're looking for is unique sequences within these 300 base pair segments. In fact, the last step, I run it against the Poplar genome because the primers could potentially amplify host DNA. And so we filter out for that at the last. Once we get it down to a manageable number, then we blast those remaining segments against the entire NCBI nucleotide database. So not just whole genomes, but partial genomes, genes, everything that's in there. And then what comes out, what remains, what I call the library of unique DNA sequences. And then we use another software, Primer 3, to design the strain-specific primers for those sequences for each genome. Then we qualify them in standard PCR in the lab because it's cheap and fast to make sure that they're specific to the target, that they don't bind off the target. For example, with the Poplar DNA or other closely related strains. Once we have a strain that we feel, a set of primers that we feel is kind of a good candidate, then we'll move it into the digital droplet PCR experiments. Now this method is not new. I took it from the two papers that are at the top. they were using the strain-specific primers with qPCR, which is another, I guess it's probably second generation, and ddPCR is probably third generation. But it's another type of PCR that you can quantify copies, but it's not, it doesn't have as a finer resolution. It's still susceptible to noise, and it requires building the standard curves, where ddPCR doesn't have that requirement. So this is the first experiment that I wanted to show you with our ddPCR. So this is a greenhouse experiment using Poplar. And this was a master student in one of our collaborators' labs. They're the plant folks, we're the microbe folks. And he was looking at the effect of stomatal patterning on a poplar under drought stress, and that are inoculated or not inoculated with the endophytes in the greenhouse. And so this was inoculated with 8 strains, but because this was our first trial, and because we were frankly in a hurry to get some data, and these ddPCR experiments they're not cheap, they're not terribly expensive, but they're not free. So we only targeted 3 strains, so 11RA, SD1, which is an azotobacter, and then a Rhinella. And you can see the concentration graph. So it's kind of hard to see on this PDF, but the first column being the control, second column being plant 33, and the top is SD1 and the bottom is WP5. You can see the concentrations about 6 copies per microliter, 6 and 7 copies per microliter. The next plant over is less than 1 copy per microliter. So that's 15 to 20 copies of our target in a single PCR reaction. Plant 2, and then plant 72. So I can already see a pattern, a colonization pattern, concentration pattern, abundance pattern by plant. It seems like 2 of the plants were more colonized and at a higher abundance. It looks like all the plants were colonized, excuse me, but few of the plants were colonized at a higher abundance than the other plants. Down here to the right in the green box is the means across all the plants in the experiment, both copies per microliter and then copies converted back to copies per milligram of plant tissue. Okay, so those numbers for copies, once we get back to milligrams of plant tissue, that's equivalent to the number of cells, the number of endophyte cells in those plant tissues. And those numbers are pretty low, right? We would expect for a microbiome to have 10 to the 6, 10 to the 5, 10 to the 6, 10 to the 7 cells per microgram. But they're there, and we were able to detect them. This is a cool paper that Matt wrote, actually. He used machine learning to estimate the changes in the morphology of the stomata and the spacing and sizing or distribution of the stomata on the leaves. And what we did is he would come and take his measurements, his physiological and his stomatal imprints off of a single leaf. I would come behind him and collect that leaf and extract the endophytes from that same leaf. This was another experiment. This one, again, using Poplar. Again, this one is a drought experiment as well. This time we measured all 8 strains in the consortium. So this is the raw data table here on the right. So you can see our different strains, and you can see by plant number the degree of colonization and the abundance. Now this is on the left. This is what's called a rhizochip, and it's a polymer that we pour in the lab, and it's got 2 pieces that come together like this, but the inside of it is patterned to mimic the pores within soil. And this was developed by our collaborators at the Northwest National Lab, so one of our national labs. And it allows them and us to look at very fine detail experiments at the root level. And so this experiment, they were measuring metabolism or metabolites in the roots and around the roots, so the exudates, under different conditions: inoculated, not inoculated, stressed, not stressed. There was some differences. I didn't look closely at the data set. There was like 6,000 columns of different metabolites, most of which they didn't know what were. But there were some that were correlated with endophyte presence. So this is kind of breaking that data down. So with this one on our metrics for colonization dynamics, we can look at colonization success, and we can look at abundance. We couldn't look at localizations because we only processed the roots. So the table at the bottom is showing the proportion of plants that were colonized by each strain in the consortium. And most of the strains colonized really well. SD1, for whatever reason, had challenges, although it did much better on the drought plants, which is DE. Well-watered is WWE. These are all obviously only looking at the inoculated ones. Okay. There was no difference well, I won't get into p-values, but you can see there's a definite trend there. There's actually, I see a trend that between the drought plants were colonized to a greater degree than the well-watered plants. Okay, so now I'm looking at colonization by abundance, colonization abundance by endophyte and by treatment. So the blue bars being the well-watered and the orange bars being the drought. Couple things jumped out at me, right? So there's a lot of variation in the abundance between the different strains. SD1 being the low end, right? And WW5 and WP5 having relatively high abundance compared to the other members of the consortium. When we inoculate the plants, they're all at the same concentration of cells based on optical density, which is not super accurate, but it's our method. So they were all introduced basically at the same concentration. This is so these numbers here, and if we look at the variance, if we look at the standard deviation, in many cases, the standard deviation is larger than the mean. So there's just a ton of variance in the abundance of the cells in the plants between plant to plant. So my next thought was to look at the variance itself, and we can see that those two strains are just dominating the noise that's in the system. And more so obviously with the well-watered plants. So then next I looked at so is there differences between individual plants? Are some plants more likely, more receptive to being colonized or more resistant to being colonized? We definitely saw some differences there. This chart, if you look at plant 27, I actually had to truncate it to be able to see the other bars, because the colonization of that plant was so high, and that's across all the strains. Whereas other plants, several of them, 42, 43, 31, 32, were relatively poorly colonized, or the abundance, I should say. I keep saying colonized, but it's really abundance. And these are copies times 1,000. So that's 1.4 million cells, which is about what we'd expect per gram of tissue. So the final, so really the final slide that I have on this project are the samples that we're collecting over a 4-year field trial that's actually ending when I get home in September. I'm going to go out and do the final collection. So the plants were planted the poplars were planted in 2022. We sampled 3 times per year. There's 120 plants. We're sampling 2 tissues. So that's almost 3,000 samples. But each sample, to run a ddPCR experiment, you have to run with the method that we're using. You have to run 1 experiment per strain. You can only detect 1 strain per experiment, per PCR. So that means that there's about 23,000 PCR reactions to do. That's not going to happen. First of all, I don't think I could get the undergrads in our lab to grind 2,800 plant samples to extract the DNA. But then it's just not cost feasible. So what I'll do is I'm going to take a subset of these and test those over the duration of the experiment. These poplars were there were several groups, 4 blocks, either nitrogen or no nitrogen, water or drought, or inoculated or uninoculated. As a matter of field research, the nitrogen's not going to work because we're actually on an agricultural extension farm of a Washington State University, which is the other big university in Washington. And of course, because they're growing agricultural products, the soil's saturated with nitrogen fertilizer. And so we're not getting a lot of response from nitrogen, but we can control the water. It's in the eastern part of our state, which is a high elevation arid region that gets 20 centimeters of rain, 20 centimeters of rain a year, so very dry. Poplars generally grow around rivers or in very wet habitats, primarily on the other side of our state where we get, I have to do the maths, but 200 to 400 centimeters of rain a year. I think that's it. I have 2 other slides that I can show from a different experiment that aren't related to ddPCR, but I think are pretty cool. I'm going to do it since nobody said no. Okay, so this paper, and the reason that I can tie this to ddPCR, to droplet digital PCR, is because, as I mentioned before, when we were adding the consortium in, we're putting them in at equal concentrations, but we know that there's interactions between our endophytes in the plant and in the solutions, in the inoculum prior to adding them to the plant. We know that there are interactions between our synthetic community, our endophytes that we're adding to the plant, and the native microbiome that exists in the plant already. And we know, of course, that there are interactions between the plant and the endophytes that we're adding. So it's a pretty complex system. So as far as an applied approach, we can use ddPCR to optimize the way that we inoculate the plants. Should they all be in the same ratio? Should they all be added to the plant at the same time, or should they be added, staggered or in stages where we add them and then re-inoculate at a later period? We can start to look at some of those questions and to be able to maximize the results for our applied projects. So this project here is looking at diazotrophic endophytes, nitrogen fixing endophytes. And we don't work, and this is a point of contention in our lab, we don't work with nodulating plants. So the traditional nitrogen fixing plant, just that term alone will make plant microbiologists sort of grumble under their breath, because it's not the plant that's fixing the nitrogen, it's the microbes, the bacteria. But the dogma is that only plants that can form legumes or other nodules in the roots, associate with nitrogen-fixing bacteria, and fix nitrogen. My PI, who has literally gotten into shouting matches at conferences with the legume people, has her idea early on, her postulates early on is that there are many plants that grow in extremely nitrogen-limited habitats, and they're not legumes. And so they have to be getting their nitrogen from somewhere. So her early endophytes that she isolated were from poplars and willows growing on a rocky substrate along a river. It spends about 7 months a year underwater. The river recedes, the poplar germinates, grows quickly, puts its roots down, and establishes on, as a primary succession, on these cobblestone riverbanks. There's no appreciable levels of nitrogen there. There's no appreciable levels of organic matter there. So her idea was, well, they must be getting their nitrogen from somewhere. And so she's isolated many free-living endophytes that fix nitrogen. And so a lot of our research has been on demonstrating that these endophytes are fixing nitrogen in the host plant and that they're contributing to the nitrogen status of the host, which would have huge implications because then we can inoculate non-legume plants and potentially reduce the nitrogen requirements, the synthetic nitrogen requirements that we're putting on the crops. So nitrogenase is the enzyme that breaks dinitrogen and begins the process of converting it into a biologically available form of nitrogen. It is very sensitive to oxygen, which is one of the legume arguments, is that the nodules create a very low oxygen, high energy environment, and that you can't fix nitrogen outside of the legumes because there's too much oxygen, and it will interfere with the process of the nitrogen. Or the nitrogenase, excuse me. So these are, what you see on the left are two examples of endophytes of ours that actually fix less nitrogen under low oxygen concentrations. So ambient oxygen's 21%. I think nitrogenase is sensitive or needs to be down below 10. It might be like 7 or 4%. I'm not 100% sure on that, but it has to be much lower than ambient. And so you can see these two strains actually produce more nitrogen, or fix more nitrogen, or excuse me, let me rephrase that. They have a higher nitrogenase activity because these are an acetylene reduction assay, which is a proxy for nitrogenase, or nitrogen, and so it isn't truly nitrogen, but it does represent the activity of the nitrogenase. Whereas the strain on the far right is probably more traditional to what you'd expect. It requires low oxygen concentrations in order to be able to more efficiently fix nitrogen. Now, one thing I want to point out is that the y-axis on these are not the same scale, right? So 11RA is fixing 10 times as much nitrogen than WP5 on the right. Okay. And the HT strains, I should mention, because I talked about the poplar strains, were isolated from plants growing on new lava beds in Hawaii. So no organic matter there. The plants have established. Our hypothesis is that if you want to find plants that host nitrogen-fixing endophytes, then you go to habitats that have low nitrogen. Okay. So these are the diagrams from the paper that we published on this. And let me back up because what we have discovered is that the diazotrophs fix more nitrogen in the presence of a non-diazotrophic helper. So we have our diazotroph strain. It fixes nitrogen when it's in pure culture in the in the acetylene reduction assay, so it's nitrogenase activity. But when it's co-cultured with a non-diazotroph, can't fix nitrogen, the nitrogenase activity goes way up. In some cases, orders of magnitude. So what this chart is showing is on the left column is what we call the helper or the synergist, doesn't fix any nitrogen. Then the next column is just the nitrogen fixing strain, but undosed, right? So it's just a control. Then the next column over is the diazotroph by itself, the activity of the nitrogenase. And then the final column is the helper and the diazotroph together. We're seeing that across many systems, so different combinations of endophyte, diazotrophs, and helpers. Although Sphingobium, for whatever reason, we haven't figured it out yet, seems to be like the ideal partner. And plants isolated from very different habitats. You have a Hawaii plant here, you have a wild poplar plant here, and I guess the other ones are probably wild poplar also, but we've got other strains that this occurs with. How we actually stumbled into this was we had originally isolated some strains and we select for nitrogen fixers on nitrogen limited media. We collect the colony, we purify it. So we thought we run the acetylene reduction assays, we record the nitrogenase activity to identify the best nitrogen fixing strains. So Andrew, who's our research scientist, the lead scientist on this, had run this assay, had the results for the nitrogenase activity in a couple strains, and then he wanted to move them over to a rich media in order to make sure that they were single pure culture strains and to continue to purify them and then make our perms, which are our working stocks that we keep in the -80 freezer. And what he realized when he streaked it out on a rich media, that there was actually 10 strains in that one, what we thought was a single strain nitrogenase fixer. So he went about his business and he isolated the diazotroph on the rich media, then ran additional acetylene reduction assays, and the nitrogenase activity dropped off the table, much lower. And so then he realized or put together that somehow the nitrogenase activity was linked to the partners being cultured together than as a pure culture diazotroph. So what we're currently working on is trying to identify the mechanism. So I think it could be related to biofilms, which are ways that bacteria or other microbes can control their environment or their habitat, mediate the stresses of their environment, desiccation, shear stress, things like that, by exuding extracellular polymeric substances, or EPS, into their environment to create a matrix or a framework around them that they then occupy. And that can create those gradients that would lower oxygen and reduce the interference by oxygen of the nitrogenase. That's one idea. They're also looking at exported metabolites and things like that. So that's really what I had. I'm interested if there's a questions.
Okay. Thank you very much, Rob. Okay, bueno, muchísimas gracias al profesor Robert Tournay y muchas gracias pues a los que están conectados y hicieron la asistencia. Si alguno tiene alguna pregunta, pues no sé si pueden abrir el micrófono, la pueden escribir acá, puede ser incluso en español, y acá yo se la transmito al profesor Tournay.
Siempre pueden abrir el micrófono los que quieran hacer preguntas, no hay problema.
Vale, muchas gracias. Carolina dice que gracias por el seminario muy interesante. Juan Manuel, esto muy interesante, gracias. Bueno, pues
Okay.
creo que no hay preguntas. Muchísimas gracias a todos los que se conectaron y posiblemente a los que después vean esta charla en la grabación donde quede colgada. Muchas gracias pues también a la Escuela de Microbiología y a todos los que nos colaboraron con la transmisión. Muchas gracias.